Hello, my name is Wayne Lam, and I'll be presenting the work that I did, sorry, the work I did making a geriatric human bone marrow atlas in the TAN lab at the Children's Hospital of Philadelphia. So, for context, bone marrow is highly diverse and it contains many different cell types fulfilling different functions. The main function of the bone marrow being to produce blood cells. And there are also many rare cell populations that are often lost during typical sequencing. For example, the mesenchymal stem cells, which differentiate into many cell types, including osteoblasts and adipocytes. And as humans age, the mesenchymal stem cell differentiation favors adipogenesis over osteoblastogenesis, which contributes to bone conditions like osteoporosis. So we employ single-cell RNA-seq and ATAC-seq to create a human bone marrow atlas and to investigate the epigenetic changes that control aging. So people might know about single-cell RNA-seq, but may not have as much knowledge about ATAC-seq. So assay for transposase accessible chromatin, or ATAC-seq, is a sequencing technique used to assess chromatin accessibility across the entire genome. Transposase enzymes fragment DNA at open chromatin regions. These fragments are purified, amplified, and sequenced like an RNA-seq. And these sequence fragments can show us regulatory sequences like enhancers, promoters, and silencers. And this gives us insight into the epigenetic landscape of individual cells and specific cell states. So to collect the cells used for sequencing, we used a workflow to enrich rare cell populations. So our cells were collected from 13 geriatric patients ranging from ages 44 to 85, undergoing a complete hip replacement. Cells from the femoral head were enzymatically digested. Red blood cells and granulocytes were depleted from all the cells. And in one-third of the cells, CD45 was depleted to isolate non-hematopoietic cells. And in another third, CD34 was enriched to isolate hematopoietic stem and progenitor cells. So all these three fractions were collected together and subjected to single-cell RNA and ATAC-seq. And you can see from this slice of the femoral head, this is our region where our cells come from. And the data was then analyzed using this workflow. Cell Ranger was used to make FASTQ files and to count gene expression genes and peaks from the gene expression and ATAC data, as well as to generate some quality control metrics. And SC Attack Pro was used to reprocess the data and to recount the peaks, as well as generate additional quality control metrics. Then low quality cells were filtered out using total UMI count between 1,000 and 40,000. Percent of reads, percent of mitochondrial reads less than 15%. Fraction of peaks in fraction of reads in peaks greater than 25%, and total unique fragments higher than 2,000. Then gene expression and ATAC data were normalized separately using standard techniques and then visualized together using weighted nearest neighbors. And then all of the samples were integrated into one data set using Harmony and RPCA, although we decided on RPCA as our final integration technique. And then finally, we annotated the clusters using the single-cell RNA-seq bone marrow atlas from Shovik et al. So our bone marrow reference comes from this paper by Shovik et al. And what we did was we projected our integrated UMAP. Oh, sorry. We projected our integrated UMAP onto this reference to be able to annotate our own cells, which tells us what our data set is made up of and our composition of cells. So here is a coarse composition of the data set. Uh, per sample, as you can see here, we have 13 samples and in total. And on the bottom is a key of the cell types and what category they belong to. So for example, for the mesenchymal stem cells, you have adipo MSCs, you have fibro MSCs, you have osteoblasts, you have osteo MSCs. And for our per sample composition, you can see that there is a lot of variation between the samples, which is definitely expected. But in total and overall, our MSC population is the highest, which means that our depletion of CD45 was successful in isolating the non-hematopoietic cells. And here we have our annotated RNA-seq and ATAC-seq integrated HuBMAP. And it shows the cell types that are present in our data set. So as you can see, similar cell types are clustered together. For example, here we have all of our mesenchymal stem cells. We have HSPCs here, the hematopoietic stem and progenitor cells. We have the T cells here. We have B cells, um, plasma cells, and also the endothelial cells, the sinusoidal and the arterial endothelial cells. And these are dot plots of marker genes for each cell type in the lymphoid and the mesenchymal stem cell groups. The dot color represents how expressed that gene is in the cell type, and the size of the dot represents in what percentage of the cells in that cell type is the gene expressed in. So this shows the top three marker genes for each cell type. And as you can see, they're pretty distinct from each other, which is good. Looking at the top marker genes for each cell type can help us come up with our own list of marker genes and validate that our annotations are correct. So on the bottom here, squared in red, are cell markers from Shovik et al. From that paper. The levels of expression with their respective cell types are pretty much expected, and it agrees with the paper. So that is a good validation for our annotations. And finally, ataxic is powerful because the peaks show how open the chromatin is per cell. These peaks represent regions such as enhancers, promoters, and silencers. So with our multi-ohm data set, we can see how exactly gene regulation relates to gene expression. High chromatin accessibility is often correlated to gene expression. For example, in our CD4 T cells, the peaks for the CD4 gene are much higher than for the CD8 T cells, which makes sense because CD4 T cells express more CD4, and the vice versa is true for CD8A, for the CD8A gene. Now, our next steps for this project are to investigate primed transcription factor sites that activate in the next cell state to identify key regulatory events that govern cell differentiation and to conduct a cell chat analysis to predict which mesenchymal stem cells are regulating hematopoietic stem cells, but also to find out what regulatory factors dictate mesenchymal differentiation and what signaling pathways are changing between different mesenchymal stem cell states. I learned oh, I learned a lot over this summer and on this project, specifically about bone marrow cell biology, as well as the typical workflow for a single cell multi-ohm data set, data analysis, as well as the statistical methods to normalize and visualize single cell data. I also learned about Seurat and Signac, which were developed by the Satija lab that Della was part of, which is really cool. And also to work with how to work with the single cell RNA-seq and ATAC-seq data using these packages, these tools. Finally, I'd like to thank the HuBMAP consortium, as well as its coordinators, my lab PI, Kai Tan, my mentor, Wenbao Yu, Danica Makowski, who helped me come to Philly and dealt with all the logistics, Michael P. Duffy, who came up with this project, and everyone in the Tan Lab for helping me throughout this project and helping me fit in. Please connect with me on LinkedIn. This is my LinkedIn, this is my email. And finally, questions?